or I have shown how you can check the quality of your data, and I've also explained how you can improve the quality of your data. So this is the part where we're going to start now. So after the quality control, we now have a very clean, fast set of FASTQ files, and this is what we're going to use for mapping. We're going to align the reads to the genome. So that means that we need to have a genome to map to. I will talk about the data formats that are generated by mapping. So it's no longer FASTQ files after mapping, but something else. And after the mapping, there are a lot of additional steps that have to be done on the result of the mapping. So I will also talk about this. And these steps are again different for DNA-seq and for RNA-seq data. I will show you how you can check the quality of the mapping. Just like you check the quality of the raw reads, you're also going to check the quality of the mapped reads. And then the nicest part of the whole training, I think, I will show you how you can visualize the results of the mapping. So that's the thing that biologists are always happy with, because for the first time they can actually see something. At this point, what we have is a clean FASTQ file. Why did we clean the FASTQ file? Well, because mapping is slow. So the more smooth you can allow it to go, the better it is. So this is why we cleaned our FASTQ files. We removed as much as possible all the things that can give issues during the mapping. So we have a clean FASTQ file. Now that FASTQ file, the only thing that it contains is the reads, the sequences themselves, the quality scores of the reads, and for every read I know which cluster it came from on the flow cell. But that's the only information I have up to now. You cannot analyze these data. Before you can analyze these data, you need to have more information about the reads. And the most important thing that you need to know about these reads is where do they come from? Which region in the genome do they represent? Which gene do they represent? So, and this is what mapping will give you, this information. So that's basically what you do here. You start from your clean FASTQ file, you take your reads, and you're going to align them to the genome. So basically very similar to what you do when you do a BLAST. There you're going to align your sequence to a whole database of sequences. Here you do the same thing, you align your reads not to a database but to the genome sequence. Now before you can do that, you need to have a genome, of course.